Horizons is about to end. And what's better than listening to one of the most interesting talks in this year's Horizons? We are happy to welcome Professor Jay Mellies with a small introduction. Jay majored in biochemistry at the University of California at Davis. After a small step outside of academia, Jay returned to UC Davis for his PhD in microbiology. With a strong interest in bacterial pathogenesis, he worked on various pathogenic strains during his 2 postdocs at the Max Planck Institute for Infection Biology in Tübingen here in Germany and the Center for Vaccine Development in the University of Maryland School of Medicine in Baltimore. Now, Jay Mellies is an Amgen Perlmutter Professor of Biology at Reed College in Portland, in Oregon, studying the molecular mechanism of flare action at promoters of genes encoding type 3 secretion system components. Most recently, Jay has performed groundbreaking research in the fight against environmental pollution. As the overall rate of recycling is less than 10% of the plastic materials made annually, with much of the waste ending up in our landfills and oceans, he and his students are investigating how biodegradation of PET, the main material out of which water bottles are made, could help to degrade plastic waste. So I think it's better to hear the story from Jay himself. Jay, we all have been looking forward to your talk, so now the screen is yours and you may start your presentation.
Thank you very much, Katerina, for that very kind introduction. I'm very excited to tell you about our work today. And as you said, I did a postdoctoral fellowship at the Infections Biology Institute in Tübingen. I was looking forward greatly to returning to Germany, to Göttingen, to this talk. And to visit old friends, but alas, we're giving the talk from offices. I'm happy to do that, and I'm looking forward to returning to Germany someday. I would also like to acknowledge you and the other students who put this meeting together. I, like other speakers who have said, it's wonderful to get invited to meetings, but it's even better to get invited to meetings that are run by students. So thank you very much for the effort in putting this all together. And as you can see from perhaps better times, I am a faculty member at Reed College, which is on the first slide here. Reed is a small liberal arts college. We only have undergraduates. We have no graduate programs, and we only have about 1,300 students here. There's a major focus on research, and so we send a lot of students on to graduate programs. So almost all of the work that you'll see today was done by undergraduates. A bit of it is published, but most of it is not yet published. We're working on that. And today I'm going to tell you a story about a student who worked with me about 3 years ago and started this new direction for me because my background really is bacterial pathogenesis and genetics. And so this is a brand new direction for us, and it's a very exciting one, where we have isolated these bacteria from polluted soils in the United States and shown that they are able to degrade PET plastic. And as Katrina says, it's a main component of water bottles. And we have a huge problem globally with plastic waste. And so this is a new direction for us. So as you know, plastics are part of our lives. They make up everything from the computer I'm looking at, the glasses you're wearing, the water bottles that we have. They're wonderful, durable materials, but that's also a curse because they don't degrade. They're highly resistant to physical and chemical degradation. And there's a lot of plastic. So since the 1950s, we've produced about 9 billion metric tons of plastic. And it's estimated that about 6 billion of those are still out in the environment, either in the ocean or on land. So on the left portion of my screen, you can see kind of some the chemical structures of some of the common plastics: polyethylene, polypropylene, polystyrene, and PVC on the left-hand side, and then PET plastic in the middle down there in the bottom, polyethyl terephthalate plastic, which is a component of water bottles. And it's a semi-aromatic polyester, which we'll talk a bit more about as we go and learn more of the specifics of the bacteria and the pathways and the enzymes that allow for degradation. All of you are aware, I'm sure, of the scope of the pollution problem. There on the screen is an image of a waste dump, and all of us have seen the heart-wrenching images of wildlife, turtles, and other species who are choked by the plastics on our planet. And so 300 million tons of plastic waste are produced every year, 10% in the US alone. And as I mentioned, about 6 million billion tons currently exist in the environment. And the scope of the problem is immense. You've all read, I'm sure, in the popular press about how there's perhaps now more plastic particles in the ocean than there are fish, which is a crazy, horrible statistic. So all of us who have the means and live in countries that have programs to do so do our best to recycle. But in working on this project, I have learned a tremendous amount about the industry. And in fact, recycling does happen, although less than 10% of plastics are ever recycled. And in the case of PET water bottles, they really are only recycled once, so very low level of recycling. An example is the Gelbesecke in Germany, of course, where people recycle their plastics and glass and other materials. But very little of it is recycled, and we certainly don't have anywhere near a circular economy with plastics. The other thing that I've learned is that plasticizers are also a huge issue. So these are chemical compounds, many of them phthalates and other toxic compounds that are placed in the materials to give them the characteristics that we want to have. So the problem is that the plasticizers make them more toxic, the plastics, and it also makes them more difficult to degrade biologically. Recently, there's been a pretty good effort to look at biological solutions to this immense problem. For example, down in the lower right, I have a reference there from the Japanese group, the Yoshida et al. Paper that hit kind of a big splash in 2016 that they were looking at a plastic dump area where they found this bacteria, Ideonella sakiensis, that was able to degrade and assimilate PET. The major effort, though, really in terms of biorecycling has been towards purified enzymes. So, for example, these alpha beta hydrolases, which is a general category. I'll talk about that more towards the end of my talk with more specifics. But in fact, these are esterases. They cleave ester bonds. And the main category of enzymes that have been found to degrade plastics are these cutenases, which are related enzymes. And the leaf branch compost cutenase is one sort of leading the pack. And what people have done really is to try to engineer these petases, if you will, to depolymerize the material so it can be recycled. So if you get down to the monomers terephthalic acid and ethylene glycol, that those two compounds can be used to repolymerize new material. Our focus has been different, though. So reduce, reuse, recycle certainly is important for all of us to follow, but there's also biodegradation. And Katrina and I were talking a little bit before the session about how I got involved in this. Like I said, I'm a bacterial geneticist studying infectious disease. And in fact, my student a few years ago, 3 years ago now, in 2017, when she took my class, Morgan Vague over there on the right-hand side of the screen, who's from Houston, Texas, read the Yoshida et al. Paper and came to me when we do an independent research project in my class, in my microbiology course, and said, hey, can we do this? And I said, of course we can do this. This is Reed College. We can pursue these different ideas. Let's do it. So for her thesis, she collected some soils from East Beach, which is near Houston. That's the map there from Google, and brought those soils back to Reed. And so the idea was Houston obviously has a lot of petroleum pollution. It's a Superfund site. There's many Superfund sites, and those are sites that are designated by the Environmental Protection Agency, United States, to be really, really polluted, and they need a lot of work to clean up. So they're both aromatic and other compounds that are from petroleum waste that are found in these areas. And so she brought it back the soil's back to reed. And so the way we began this odyssey was to look for lipase activity, because lipolytic activity is basically cleaving ester bonds, and some of the structures of the polyesters are similar to lipids. And so we initially screened for lipase activity with the idea that these bacteria might be able to degrade PET plastic. So the arrow down there on the right is a simple agar plate with this dirhodamine B, that when the fatty acids get cleaved off the triglycerides, they fluoresce under UV light, and you can look at the image there. And so she found these bacteria that were able to degrade the substrate, which in this case was olive oil. So olive oil on the plate, and you can see the fluorescence down on the bottom right over here. So on this plate, we have basically the 5 strains that we isolated, 3 pseudomonads and 2 bacillus species, and an E. coli control. So here is just plain LB agar or nutrient agar with E. coli control and the strains. And if you shine UV light on this simple agar, you can see a little bit of natural fluorescence of the pseudomonads, but the halos here are indicative of lipolytic activity. So rhodamine B fluoresces then when the fatty acids are cleaved, and so they bind to the fatty acids and fluoresce. And so you can see this nice halo for this isolate, and this isolate. And so we isolated then 5 strains as part of 2 consortia. And so we sequenced the genomes of these, and that was published in Microbial Resource Announcement, which is a publication by the American Society for Microbiology. And so our 5 isolates were consortium 9 contained isolate 9.1, which was a Bacillus thuringiensis, 9.2 Pseudomonas. We had a second consortium 13 where these strains grew together, Bacillus and Pseudomonas, and then a single isolate of Pseudomonas. While these two by 16S analysis, 16S ribosomal RNA analysis were the same species in the database, they in fact have quite a number of genetic differences after you look at them. So the lipase positive were Pseudomonad species, the lipase negative were Bacillus, so it's a simple gram stain, pink for gram negative, purple for gram positive. And to signpost ahead a little bit, I found it really endearing that there was precedent in the literature for these 2 bacterial species to be friends. In fact, they've been isolated as consortia previously, and I'll tell you that bit of information more specifically as I get to the end of my talk to tie together how the bacteria actually are cooperating to degrade the PET plastic. So the first thing we did basically was to just grow the bacteria on PET plastic. And the question was, can you use a carbon-free medium that just contains buffer, pH 7.4, some metals, and you put in PET plastic as a sole source of carbon, can the bacteria grow? And the answer, of course, is yes. Otherwise, I wouldn't be sitting here giving you this talk. So as we found that the consortia or the groups of bacteria together grew better than the individual consortia or the individual isolates. So this is post-consumer PET, which is basically water bottles, and then granular PET. So this is a material that you can buy, for example, from Sigma, and it's a more pure product. And so the bacteria are able to grow better. So we were measuring just optical density or absorbance at 600 nanometers over time, over several weeks, of course. And so Cam Roberts picked up this project after Morgan graduated, and she collected these data. So you can see this is the full consortium here. There's a steeper slope here, which indicates faster growth and a higher growth yield for the consortium versus the other individual consortium. I have consortium 9. Consortium 13 is not on the graph here. It's similar to consortium 9. I took it off to make it a little simpler for you to look at. One thing that was really interesting to us that this strain, in fact, 9.1, which is a bacillus species, couldn't grow on PET plastic by itself. However, consortium 9, which contains both the Pseudomonas and the bacillus, can grow. So somehow they were cooperating. We use this. You can actually plate the bacteria out using different antibiotic resistances, because often Pseudomonas species are resistant to a lot of different antibiotics. And so we knew that actually in consortium 9, which is here, it contained about 50% of the Pseudomonas and 50% of the Bacillus species. So they're growing together. So that was very interesting. And so in the literature also, we knew that, for example, often bacteria will grow on the surface of the plastic. And in fact, biofilms are associated with the degradation of plastics, different types of plastics. And so this slide is just some data that Cam collected as well to show that the vast majority of the bacteria growing in the culture were actually growing on the surface of the plastic as opposed to in the medium. So here's 9.1. It didn't grow. You can see 9.2, which is a Pseudomonas. The majority of the bacteria were growing on the surface of the material as opposed to the media. And that was true for the granular PET as well as the post-consumer PET or the water bottles. So on the surface. As I mentioned, there is precedent in the literature for showing the biofilms are associated with plastic degradation by bacteria. And these are just some initial images on post-consumer plastic where you can see the bacteria in groups or in clusters on the surface. And if you look more closely, you can actually see in this image, for example, in B, this goopy stuff we think is polysaccharide. And extra polysaccharide is a component necessary for biofilm formation. So this is what the arrows are pointing to here. These groups of bacteria, the scale here is about 5 micrometers. Pseudomonas about 1 micrometer in size or so. A little closer in, then you can see with the consortia, not only the extra polysaccharide, but you can also see these little fibril structures, which are most likely pili. So the surface of the plastic is very hydrophobic, of course, and hard for the bacteria to grab a hold of. But in fact, the plastic or the pili here are also quite hydrophobic, and they allow the bacteria to adhere to surfaces. You can also use scanning electron microscopy for looking at the degradation. So sort of standard in the field that you remove the bacteria off the surface of the plastic, and then you can take a scanning electron microscopy image of it to look for some sort of surface degradation. So that's what we did. This actually happens to be amorphous PET, which is a little less crystalline and it's more easily digested. So the bacteria were incubated for about 4 weeks, 30 degrees Celsius in this carbon-free media with amorphous PET. And these are the consortia, so the Pseudomonas and the Bacillus strains together. On the top panel here, these pieces of plastic were treated with a mild detergent, saponin, which is going to remove the biofilm. But what we found was that it was really difficult to remove all of the bacteria from the surface. So you can see here these cute little pseudomonads are on the surface of this plastic. And it even looks like in some of the images that we can see that they're kind of embedded within the surface. So with the shadowing, it's kind of hard to tell, but that's kind of what it looked like. You can also see with the Bacillus species, and of course, the Bacillus and one of the Bacillus at least, and all of the Pseudomonads can grow on the PET plastic by themselves. So you can see the Pseudomonads, which they tend to grow in these streptobacilli or these chains where they seem to also be embedded within the plastic. This here, interestingly, if you look in the literature, probably is a piece of crystalline PET, where this is less crystalline here. So this portion of the material is obviously more difficult to degrade for the bacteria. So over here is the control where it's not incubated with black bacteria, but rather it was treated. So you can see, the bacteria are really tightly adherent to the surface. The bottom panel then is where we've actually treated them with proteinase K. So the proteinase K actually degrades the pili or all the other proteinaceous structures on the bacteria that allow them to adhere to the surface. And what you can see pretty clearly is this set of holes in the material. And this is kind of reminiscent of incubation with a purified cutinase, where the cutinases do create these holes. So it looked like it was consistent with what we'd seen in other papers. over here in this image, you can kind of see this bowling pin. This looks like some sort of dead bacillus here, and the bacilli are a little bit bigger. They're about 5 micrometers in length, and what we noticed was there's some sort of trough-like structure here that looked like there is alteration of the surface of this material. So it looked like, I don't know, I don't want to make too much of it, but the bacillus seemed to make these sort of trough-like indentations, and the Pseudomonas, or perhaps secreted enzymes from the bacillus as well, could create these hole-like structures. So it was evidence, anyway, that the surface of the material was being modified by the bacteria. So looking further, we wanted to gain some additional information, or additional evidence rather, of modification of the surface of the plastic. So this is Fourier transform infrared spectroscopy, and basically you can look at different functional groups. And so here's the pretty well-known pathway for degradation of PET. Basically, PET is cleaved into bis hydroxyethyl terephthalic acid to mono, and then that is cleaved into terephthalic acid and ethylene glycol. And so you can see then you can have more hydroxyl peaks as, for example, the PET gets degraded. We observe an increase in hydroxyl peaks. Those are over here. Also, aliphatic peaks increases, and the carbonyl peak as the plastic gets degraded is going to decrease. And so here are the ester and the carbonyl peaks that decreased, and the control here is a culture that contains just E. coli. And so it's quite different because E. coli, of course, can't degrade the PET plastic, but the functional groups on the surface of the plastic are being altered. So that was further evidence that the bacteria are actually degrading the plastic. This assay Cam did during her thesis year, and this one, as opposed to growing bacteria on the plastic, what Cam did was she initially grew the bacteria on BHET, and then she harvested the supername. She spun the bacteria out. So these are they're not purified, but just the enzymes that were found in the growth media she placed on BHET and monitored then the presence of TPA using NMR. And so these data are presented here. And so what was really fascinating to us is that by the secreted enzymes, that only the Bacillus species 9.1 and 13.1 alone were able to create significant quantities of TPA, or show significant cleavage of BHET by secreted enzymes. Of course, the consortia, interestingly, were able to do that to a much greater extent, both Consortium 9 and Consortium 13. But the full consortium, because these were normalized to a peak that indicated BHET degradation, there was no BHET left. Basically, the full consortium fully degraded the BHET. So it was completely degraded. And so we're presuming that there was a large amount of TPA, but we couldn't really normalize and put the quantity here because there was actually no BHET left, which was really pretty interesting. So, of course, if you're going to address plastic degradation on a large scale, you need to be able to show that there's a change in weight. So if you take a kilogram of plastic, can you show degradation over time? So this is some work that Cam did as well. So what she did was she incubated in the carbon-free media with plastic, in this case granular PET. The different isolates were grown for basically 28 days. And so you can see then that the full consortium degraded the plastic by about, in weight by about 3% over this time period. So that was greater, which is consistent with our previous data, than any of the individual consortia or the individual isolates together. And of course, 9.1 was not able to degrade. And this is in culture, of course. This is not purified or partially purified enzymes. So this brings me to the question, how are the bacteria acting synergistically? This is the novel portion of our work, really, because we've known that the bacteria can degrade plastics since the 80s. but them working together in these consortia is kind of a new idea. And so, as I mentioned to you as a kind of a teaser at the beginning of my talk, that there's precedent for these two bacteria being friends out in the environment. So in this particular paper that I have the reference for here, some Japanese researchers about 20 years ago found that that Pseudomonas and Bacillus together in a mixed culture can degrade terephthalate found in wastewater. So plastics obviously end up on our wastewater. TPA is a degradated product, and it's a huge problem. And so they found that these bugs work together. So there's precedent for them to be friends. So following up on that, research associate in the lab, Sabrina, basically what she did was sort of repeat the rhodamine B olive oil assay to look for different hydrolytic activities. So basically this large category of enzymes that cleave ester bonds have different substrates. And so we wanted to look and see what they might be able to do. So tributylin is a simple triglyceride, and all bugs that we had could degrade that. If you look over here to the olive oil, this is degraded in the rhodamine B assay by the Pseudomonas species, whereas we had sort of differential abilities of the bacteria to degrade triglycerides with different fatty acid chain lengths. So these are sort of medium to longer fatty acid chains. Polycaprolactone is a polyester, which has a much longer molecule, of course, and only the Bacillus species were able to degrade the polycaprolactone. So that was very interesting to us. It says that when starting to look in the genomes of the organisms, there are a lot of different types of enzymes that can degrade polyester bond or ester bonds that we really don't know the full extent of what they can do. So these enzymes are really part of a larger category of alpha beta hydrolases. They have this typical catalytic triad, which include serine, histidine, and aspartic acid. And on the structure, this is a predicted structure of one of the esterases that we have in the genome of 9.2, which seems to be somewhat of our superstar degrader. So that is found in this active site here. But there are a number of different enzymes in this group. Lipases, cutinases, as I mentioned, esterases, which tend to cleave triglycerides with shorter fatty acids, and then carboxyl esterases as well. And so some of the bioinformatic work and the structure prediction was done by actually a rising junior, Riaz Ditter, this summer working with me. So what are the possibilities for PET cleavage? We really don't know the initial enzymes that are cleaving PET, although there seem to be enzymes in the genomes, encoded within the genomes of both of these organisms, or both of these genera, if you will, that might be doing the work. So certainly there's polyesterases because the Bacillus species can degrade the polycaprolactone. And a couple of suspected hydrolases for PET degradation. So there's an esterase, which is predicted to be secreted into the extracellular area by the sec-dependent mechanism, doing some bioinformatic work. And Henry this summer did some of this, did some protein structure prediction. For example, there's a type 5 lipase encoded within the Pseudomonas genomes. And this one's interesting to us because it actually is associated with the outer membrane. So this is a beta barrel structure which embeds the molecule into the outer membrane. And here's the catalytic domain here that sticks out. And we think maybe this might play a role because of lipases needing to be able to degrade the triglycerides really at the hydrophobic, hydrophilic interface, that this might help the bacterium do that, and it might allow it to stick to the structure strongly, as you saw from the electron micrograph. So moving to the next molecule, of course, BHET, only the Bacillus really had this enzymes that are able to be secreted and cleave BHET. But as you saw, or from the graph over here, rather, that we knew that the BHET was able to be grown on as a sole carbon source by all of the strains, including the pseudomonads. So perhaps the pseudomonads aren't secreting an enzyme to degrade BHT, but they may be taking up the material and degraded it intracellularly. And again, we found that cleavage was better within the consortia. TPA degradation. Interestingly, only the pseudomonads contain the genes necessary for TPA degradation. And terephthalic acid basically gets funneled eventually through the TCA cycle, through acetyl-CoA and succinate, and it's taken up. And we have a couple of putative transporters that we think it's bringing the molecule into the cell. No TPA metabolic genes were found in the Bacillus species. So in order to degrade TPA, of course, we have to have the pseudomonads as part of the consortia. Ethylene glycol degradation is mixed, and there are about three main pathways for ethylene glycol degradation in bacteria, and we're trying to tease out which ones our bugs have, but basically ethylene glycol can be taken up and used as a sole carbon source. All of our strains can grow on ethylene glycol as a sole carbon source, so most likely it's taken up and degraded and used to produce new cells and energy, of course. So, in conclusion, we've isolated 3 pseudomonads and 2 bacillus species that came from petroleum polluted soils in the Houston area. Because of the petroleum industry, there's a lot of pollution there. We initially screened for lipase activity, and taking from the literature that we knew that could be associated with plastic degradation. Specifically, we have evidence for PET degradation, which includes the spectroscopy, as well as scanning electron microscopy images. We do have some NMR as well. I didn't have time to show you those data, but I showed you the secretion data. And also 3% reduction in weight of the PET, granular PET over 8 weeks incubation. So I think the takeaway message then is, and what's novel about our work is the full consortium grows faster to higher yields on PET plastic as well as BHT than the individual consortia or isolates. And I want to emphasize the synergy, and I think this is important for a number of different reasons. So firstly, the bacteria are working together, the pseudomonads and the bacillus species. But as I put this little image over there on the right-hand side, we as scientists need to work together. We need to work together and solve the world's problems. And there are no borders in science. I'm very happy to be at this conference. I feel very honored to give this talk to all of you today. But let's work together. We need to do this moving forward. So, lastly, I'd like to do some acknowledgments. I've had quite a number of students who have worked on the project, beginning with Morgan and through this past year and moving forward. I've also had a number of great collaborators. Rosa is at Willamette University, just south of us, who's helped us with the bioinformatics and genomics. David Payton and his postdoc in a dual over Portland State in the chemistry department has helped us with the NMR. Research associates as well as individuals in the chemistry department here, funding from the National Science Foundation. And Morgan has a TED Talk on isolating these bacteria. It's had over 2 million views. She did a really great job of explaining the project. So if you're interested, I would encourage you to take a look at that. And lastly, thank you very much for your attention today. I'd be happy to take questions.
So, Jay, thank you very much. I think your talk was very interesting, and I believe everyone in the audience is already thinking of ways how can we actually use this bacteria. Well, I will go through the questions, starting from the most upvoted ones. So, a question from Florian. Are the bacteria actually capable of completely degrade the plastic, also under non-laboratory conditions? And how much time is really needed for this? Is there a chance to distribute potent cells to new habitats to clean microplastics without in other ways destroying environments?
Yeah, there's a lot of questions there. So firstly, can they completely degrade the plastic? No, not yet. Not that we've seen. We see 3% over 8 weeks. The crystalline material is very difficult to degrade. Really, I think we have some carboxylesterases and other enzymes. I don't know exactly what all our bacteria can do, but I don't think it's going to be in my lifetime or your lifetime. That's going to happen. So under different laboratory conditions, perhaps we, as you saw in my slide, pre-treated the plastics with UV light. That creates carbonyl groups and other functional groups that are able to be more readily degraded. So perhaps with pre-treatment or other ways, other means of compromising the plastic structure, we can get the bacteria to fully degrade the plastics.
So our next question is from Professor Ram Krishnamurthy. Do these bacteria, are they fed with these plastics that are fed with these plastics show any signs of addiction? That is, are they robust in going back to their normal diet after being exposed to the plastics?
That's a good question. We haven't actually done that experiment. I'm assuming that over time they would perhaps lose the genes necessary for sort of normal metabolism. Although pseudomonads, as probably most of you know, are extremely resourceful. They have large genomes because they can degrade a lot of different aromatic and aliphatic compounds. So they are resourceful. I would imagine that most likely they could go back to a quote unquote normal diet after being propagated on a plastic. But like I said, we haven't done that experiment.
Thank you. The next question is from me. Although you talked about it, what is your experimentally described time scale of degradation? But there is another question. So with PET as a sole nutrient source, we saw that the bacteria reached the exponential phase after 20 days of incubation. So is there a chance that they show some preference over glucose in the case of PET waste that is not as pure? So probably there would be some pre-treatment needed in PET itself so that the bacteria are actually capable to show preference over PET degraded.
No, that's an excellent question. Thank you, Katrina, for that. So basically, there's long-standing of what's termed auxotrophic growth. So bacteria are able to switch from one nutrient source to another one. The classic example of that is in E. coli, and they can preference using glucose over lactose, right? That's the whole reason the lactose operon, the lac operon, is regulated the way it is. So the bacteria are going to preference using a more friendly carbon source like glucose or whatever it is over using the PET plastic because it's more difficult. We have conditioned the bacteria by growing them and dilute nutrients. So for example, you can place them in a solution with dilute nutrient agar or yeast nutrient. And once they run out of that, then they will begin to degrade the PET, right? So you can condition them, get them to grow, and then they will begin to grow on the PET plastic because they have nothing else to grow on.
So maybe I can have a follow-up question. Have you tried to genetically manipulate the bacteria in order to show preference to PET, maybe by deleting some genes encoding for enzymes for taking over glucose or for using glucose as a source?
No, that's a good idea. I think that also you can do what's called I'm not sure if folks are aware of it, but adaptive evolution. That you can basically allow the bacteria to adapt to new conditions and to get them to grow a bit better. I hadn't thought of actually deleting genes of basic pathways like glycolysis. We don't need that glycolysis thing, right? Just wipe it out. Just have them, feed the TPA and ethylene glycol through the TCA cycle. And in fact, pseudomonads are respiratory organisms. They don't ferment, so that might be a good approach.
Maybe they won't like it that much, but anyways. So next question is from Ilvika. What are the effects on environment that these bacteria can have post degradation of plastic? Do these bacterial cells undergo apoptosis?
They go undergo apoptosis. Is that what you asked?
Mm-hmm. So
Uh,
after they degrade the plastic, do they die?
uh, they will die over time. Bacteria will get into most bacteria will have adaptive means to stay alive, right? So in theory, if you get the bacteria to completely degrade the PET plastic, all you're going to have left is a bunch of bacteria, right? And it's like any other carbon source. You are going to release CO2, which some people have said, well, that might not be a good thing in itself. But so, for example, bacillus species form spores, right? So there's four spore formers, and so they can. I mean, who knows how long spores can last? I mean, uh, people have isolated fossilized spores that are millions of years old that they can, reconstitute in the laboratory. Pseudomonads can't do that. They will die over time, although they do have ability to, be viable for a certain amount of time after they grow on the plastic.
Thank you. So maybe one last question. So regarding the future application of PET eating bacteria, which one do you think would be the best approach? We just release the bacteria in the environment with a possible disturbance of the environment itself, a controlled degradation of pre-treated PET waste in special facilities, or maybe purification of the enzymes that degrade PET and treatment of PET waste.
Yeah, excellent. I would say all but the first one. So I think that I've always thought that the degradation should occur in some sort of controlled environment. I think it would be detrimental, for example, at least where we are today, to take the bacteria and take them out to the gyres in the ocean or onto land where there's PET pollution and to just cut the bacteria loose. As you say, and all of the audience knows, I'm sure that microplastics are really horrible for the environment. And so I wouldn't want to have it where just some of the plastic gets degraded and we just leave little bits everywhere. I've always envisioned having some sort of controlled environment, whether that be the bacteria growing on the plastics or purified enzymes to depolymerize them to make new products, to make a more circular economy. I think that all of those ideas need to pursue. It's such an enormous problem. I think everything we can muster and throw at this problem, we need to do it. Whole bacteria, adaptive evolution, make them degrade the plastics faster, purify enzymes. There are a lot of people who are trying to work on this problem because it's a very difficult one. And I think we need to all work together and have a lot of different approaches.
That's great. Thank you very much for your talk and for being with us.
You're welcome.
Uh, so I would like to remind to our participants that later on it's the awarded student talk of Anna Rutnunian. It's at 7:30. And afterwards, we have our social event where we meet the participants for a Horizon Sunrise. So please stay tuned and join us for the rest of the day. So, Jay, thank you very much again.
You're welcome. Thank you for organizing.
Bye, everyone.
Bye bye.